Insect growth regulators, natural hormones of insects, insect pest control, IGRs used in pest management, JH mimic, chitin synthesis inhibitors, benzoyl phenyl urases, then ecdysone agonists, pros and cons of IGRs. Insect growth regulators. How many slides we have got? 18. Okay. So insect growth regulators, IGRs, are compounds which interfere with the growth, development and metamorphosis of insects. IGRs include synthetic analogues of insect hormones such as ecdysteroids and juvenoids and non-hormonal compounds such as prescenes, anti-JH, and chitin synthesis inhibitors. Insect growth regulators disrupt and impede the development of life cycle of insects in a larval and egg stage, in the larval and egg stage. In short, IGRs is a form of birth control for pests which helps keep the population of unwanted pests under control by preventing current and future infestations. Natural hormones of insects, brain hormones. They are also called activation hormone. AH is secreted by neurosecretory cells. NSC, which are neurons of central nervous system, CNS. Its role is to activate the corpora allata to produce juvenile hormone. Juvenile hormone, JH, also called neotitinin. It is secreted by corpora allata, which are paired glands present behind insect brain. Their role is to keep the larva in juvenile condition. JH1, JH2, JH3 and JH5 have been identified in different groups of insects. The concentrations of JH decreases as the larva grows and reaches pupal stage. JH1, 2 and 6 are found in larvae, while JH3 is found in adult insects and are important for development of ovary and adult insect females. Ecdysone, also called molting hormone, MH. Ecdysone is a steroid and is secreted by prothoracic glands present near prothoracic spiracles. Then molting in insects is brought about only in the presence of ecdysone. Ecdysone level decreases and is altogether absent in adult insects. Then insect pest control. A new approach to insect pest control is the use of substances that adversely affect insect growth and development. These substances are classified as insect hormone mimics or insect growth regulators, IGRs, owing to their effects on certain physiological regulatory processes essential to the normal development of insects or their progeny. They are quite selective in their mode of action and potentially act only on target species. Inaction of IGRs, however, should not be confused with other synthetic insecticides such as organophosphates and carbamates, since these chemicals interfere with other physiological processes but do not regulate the development of normal insects. In IGRs, therefore, it does not necessarily have to be toxic to its target but may lead instead to various abnormalities that impair insect survival. Most of the IGRs that have shown effectiveness against insect pests cause the rapid death of the insect through failure of a key regulatory process to operate or function. IGRs generally control insects either through regulation of metamorphosis or interfere with reproduction. IGRs used in pest management, ecdysoids. These compounds are synthetic analogues of natural ecdysone. When applied in insects, kill them by formation of defective cuticle. The development processes are accelerated by passing several normal events resulting in integument lacking scales or vaxillae. Then juvenoids, JH mimics. They are synthetic analogues of juvenile hormone, JH. They are most promising as natural hormonal insecticides. They are most promising as hormonal insecticides. JH mimics were first identified by Williams and Slama in year 1966. They found that the paper towel kept in a glass jar used for rearing a Pyrrhocoris bug caused the bug to die before reaching adult stage. They named the factor from the paper as paper factor or juvabione. They found that the paper was manufactured from the food pulp of balsam fir tree, which contained the JH mimic. Juvenoids have anti-metamorphic effect on immature stages of insect. They retain status quo in insects. Larvae remain larvae and extra supernumerary moltings take place, producing super larvae, larval pupal and pupal adult intermediates which cause death of insects. Juvenoids are larvicidal and ovicidal in action and they disrupt diapause and exhibit embryogenesis in insects. So methoprene is a JH mimic and is useful in control of larvae of hornfly, store tobacco pests, greenhouse homopterans, red ants, leaf mining flies of vegetable sunflower, anti-JH or precusins. They act by destroying Cocora lata and preventing JH synthesis. When treated on immature stages of insect, they skip one or two larval instars and turn into tiny precocious adults. They can neither mate nor oviposit and ISO. EMD, FMEV and PB, peperonyl butoxide. Chitin synthesis inhibitors. Benzoyl phenylurase have been found to have the ability to inhibit chitin synthesis in vivo by blocking the activity of the enzyme chitin synthetase. Two important compounds in this category are diflubenzoin and penfluoron. The effects they produce on insects include disruption of molting, displacement of mandibles and labrum, adult fails to escape from pupal skin and eyes, oviductal effect. Chitin synthesis inhibitors have been registered for use in many countries and used successfully against pests of soybean, cotton, apple, fruits, vegetables, forest trees and mosquitoes and pests of stored grain. Chitin synthesis inhibitors, the insect cuticle, serve as an interface between the living animal and its environment and forms the exoskeleton supporting the lining of the gut, respiratory systems, reproductive ducts and some gland ducts. It consists of protein and chitin fractions. CSIs interfere with the polymerization of chitin. This mode of action has been targeted for control of several different insect pests. CSIs cause abnormal deposits of endocuticle that accumulate during molting, specifically uridine diphospho and acetyl glucosamine monomers, thereby preventing chitin synthesis. This produces a weakened cuticle and causes mortality when procuticle is subjected to the stress of ecdysis and cuticular expansion. Consequences of CSI toxicity also include mortality in the absence of metamorphosis and include swollen appendages, decrease in locomotion, inability to eat due to dislocation of mandibles, malformed or absent peritrophic matrix, as well as suppressed fecundity and egg viability. Benzoyl phenol urea, it's a important type of insect growth regulator acting on the larval stages of the Lepidoptera insect by inhibiting chitin synthesis have been rapidly developed after the production of the first benzoyl phenylurea diflubenzorin in 1972. Benzoyl phenylurea have a unique mode of action coupled with a high degree of activity on target pests and low toxicity to non-target organisms. That is why they have attracted considerable attention for decades and have become a new tool for integrated pest management. Benzoyl phenylureas. The in vivo or in situ isolated integument incubated in a tissue culture medium studies showed that chitin synthetase catalyzes the incorporation of UDPN, acetyl glucosamine, to chitin was clearly inhibited by BPUs. The ecdysone agonists, dibenzonyl hydrazine (DBH) compounds are non-steroidal activators of ecdysone receptor complex in insects. Their use as insecticides is based on their capability to produce a premature and incomplete molt during which susceptible insects die from desiccation and starvation. Ecdysone agonists. DBH compounds can be applied to the field because of their favorable physicochemical and biological properties such as high hydrophobicity, which allows the penetration of cuticle and the midgut of insects and their slow clearance from the insect's body, resulting in persistence of action. In addition, DBH have high safety profile which is compatible with their use in integrated pest management programs. All commercial ecdysone agonists used in IPM are DBH analogs such as tefufenozide, methofenozide, halofenozide, and chromafenozide. The ecdysteroid agonists represent a new class of IGRs which has been developed to provide safe insecticides with high activity in Coleoptera. DBH compounds are considered to be highly specific to Lepidopteran insects, although halofenozide is also marked for control of Coleopteran pests. Pros and cons of IGRs. Advantage of using IGRs: effective in minute quantities and so are economic, target specific and so safe to natural enemies. Biodegradable, non-persistent and non-polluting, non-toxic to humans, plants. Disadvantages: kills only certain stages of pests. Wait a minute. Effective in minute quantities and so are economical, target specific and so safe to natural enemies. Biodegradable, non-persistent and non-polluting, non-toxic to humans, animals and plants. Disadvantages: kills only certain stages of pest. Slow mode of action since they are chemicals, possibly to build up of resistance. Unstable in the environment.